I'm really excited to be able to talk about an ecosystem that I just really love, longleaf pine savannas in the southeastern United States. This is stemming from a line of research that I started as a postdoc, it's like 10 years ago now, and has developed into one of these large-scale experiments that Lauren mentioned, taking place in South Carolina. Okay, so before I get started, I have to say my thank yous. I'm going to be talking about work that's been a collaborative effort across a number of different researchers over the last 8 years or so. So among those are Nash Turley, whose work I'm going to feature prominently today. He's a now former postdoc with my group and is now postdocing at University of Central Florida. And Joe Litvina, who's our now former technician, now works for NEON. John Orrock is our collaborator from the University of Wisconsin. He's on the faculty there. And so it'll be work that's collaborative with these three that I'll be mostly talking about today when I'm talking about restoration for plant communities within this ecosystem. At the end, I want to touch on some of our ongoing work that focuses on restoration for pollinators, pollinator communities, and pollination within this system. And for that, we're collaborating with some familiar faces here on campus, Rufus Isaacs, who's in entomology, Jason Gibbs, who was recently in Rufus's lab and is now at the University of Manitoba on the faculty, and then also our technician now leading the project and helping to lead these efforts, Sabri Breland, who's based at our research site in South Carolina. I also need to thank the U.S. Forest Service. They are our cooperators and collaborators. None of this would be even remotely possible if it wasn't for the work that they do on their end to oversee a lot of the logistical aspects of the research, including the implementation and maintenance of our experiment. Number of people to say thank you to, and also to the Forest Service at Savannah River Site for assistance with providing funding. Okay, thank yous out of the way. So the work that we focus on in our lab and that I'll be addressing today confronts issues of land transformation, which collectively are considered the greatest threat to the world's biodiversity. And the numbers are just really pretty staggering. We estimate that on the order of half the Earth's terrestrial surface has been substantially transformed for one type of human land use or another. These span livestock production, conversion to silvicultural systems, conversion to a wide variety of agricultural systems, and of course, conversion to the places that we live and take up residence. So again, this is considered collectively the greatest threat to the world's biodiversity. And the question then is, what are we going to do about this? So preservation is got to feature really prominently here, conserving those high quality natural areas that remain, but it's becoming increasingly clear that preserving land alone is not going to be sufficient for achieving our conservation objectives in many landscapes, particularly highly transformed landscapes, not so uncommon like the ones that we have around us here in the Midwest. And so what's clear is that restoration is going to have to be part of this solution. Through restoration, we can increase the amount of habitat and the quality of habitat for all the different sorts of plants, animals, and other critters that we care about in our conservation objectives. So what do I mean by restoration? So by restoration, we are doing intentional activities as humans to cause ecosystems that have been damaged or altered, typically by some of our other activities, so that they better possess the biodiversity or the ecosystem functioning that would be representative of what we might call a natural range of variation for that particular ecosystem type that we're looking to restore. So as way of one example for the ecosystem I'm going to be talking about today, longleaf pine savannas, here's what this process might look like. So we have a forested area. This is actually a former agricultural field. So decades in the past, this was a tilled field supporting some sort of annual cropping system. It was subsequently abandoned from agriculture. Trees were planted onto the site. And so we have now this dense sort of forest in an area where we might want to restore an open canopy savanna. So what's this process look like? So perhaps we first get in some harvesting equipment. We need to thin out the trees on this site so that it's got the architecture of a savanna, making it look something like this. We've gotten the tree spacing right. We may or may not want to add species of plants or even some species of animals into these sites. And then we need to maintain this site using prescribed fire, which is a natural disturbance in this ecosystem. So this is just a very coarse overview of one example of ecological restoration. This is the practice of restoration. The science of restoration is restoration ecology. That's what we focus on primarily in my lab and what I'm going to be talking about for much of the talk today. So the science of restoration ecology provides tests of restoration strategies, conceptual guidance to this practice of ecological restoration. So a little bit on the goals of restoration before I get into the specific system I'll be talking about today. So in restoration, we, again, typically want to cause sites that we would call degraded, that have been altered by some sort of human activities, disturbances, so that when restored, they may better possess this range of characteristics, often measured in terms of aspects of biodiversity or ecosystem functioning, increasingly maybe ecosystem services, which are underpinned by both biodiversity and the functions that natural ecosystems provide. A very common way we might measure this would be to compare this restored site to, well, what we started with, the degraded site, and or to what we call a reference site, a pristine example, or ideally many pristine examples of that particular ecosystem type. So how do we do? There have been a set of meta-analyses that have been coming out that have been compiling this wide range of restoration studies that have often been case studies in particular ecosystems. And as a generality, what we've seen is that restored ecosystems have higher levels of biodiversity, higher levels of ecosystem functioning and services compared to degraded sites, but they fall short of what we see in reference sites. And so we're kind of, as a field, middling a little bit. We're having some success, but in some cases we also have failures, and we see wide ranges of variation in the outcomes that we may class ranging from success to failure. So this sets up some important questions for the field of restoration ecology. Why do we see this variation? What is it about particular sites and particular restoration approaches that may lend themselves to being relatively more successful or less successful? And why is it that our restorations, at least on average, are not approximating the conditions that we see in intact examples, intact reference sites? So what I would argue is that what we need are strong experiments where we can draw cause and effect conclusions and dig into some of the mechanisms that cause restored sites to have the characteristics that they have, and that cause, in turn, those restored sites to have biodiversity or functioning different from degraded sites and different from reference sites, and thereby we can feed that information back into restoration practice, hopefully inform better restoration practices that helps to minimize that discrepancy in the conditions we see in restored and reference conditions. So these are the sorts of ideas that I want to be talking about today in context of this focal ecosystem, longleaf pine savannas. Who's been in a longleaf pine ecosystem before? A few people here. So longleaf pine savannas are part of a broader southeastern coastal plain set of ecosystems that have been recently recognized as a world biodiversity hotspot. Longleaf pine systems, so these are systems that have a major, if not dominant, overstory component of longleaf pine trees, span from Texas to Virginia down into peninsular Florida. Historically, this was tens of millions of hectares on the southeastern coastal plain, which is the portion of our country that when oceans were higher were the bottom of the ocean. These are extremely sandy soiled ecosystems, 90+% sand fractions in the soils. They're excessively drained. They tend to be really droughty systems in spite of the fact that they do have sufficient rainfall or quite high amounts of rainfall, not that different from what we experience around here. Fantastic biodiversity in these systems. So I mentioned that they were recently recognized as a world biodiversity hotspot. Some reasons for that are in terms of the high levels of species diversity and endemism. Over 900 species of endemic plants alone in the southeastern coastal plain. So those are plant species that occur here and at least naturally outside of human transport, nowhere else in the world. Some really neat biodiversity in pictures. Here's the Venus flytrap, which is one of those endemic plant species native to the southeastern US and longleaf pine systems. Gopher tortoises, so these are a burrowing tortoise. They dig these burrows underground. When they abandon these burrows, those become habitat to many, many dozens of species that are completely reliant on those abandoned burrows, including the indigo snake, which is the longest snake in North America. incidentally. So when the gopher tortoise goes, the indigo snake goes as well. Red-cockaded woodpeckers, those are a federally endangered woodpecker species. They create their cavity nests in living, usually longleaf pine trees, which makes sense when you consider the frequency of fire in this ecosystem. Fires burn through this system on average every 1 to 3 years, and you can imagine the consequences if you live in the typical habitat for woodpeckers, at least around here, standing dead trees, which would be tinderboxes and those sorts of situations. Now, one important point about all of these species, the vast majority of these species that are specialists in this system, is that they're reliant on these fire-maintained conditions. So the longleaf pine savannas are characterized by relatively widely spaced longleaf pine trees, frequent fire, which burns the litter and herbaceous layer, again, every 1 to 3 years. And that frequent fire then maintains the structure in the system, prevents it from growing up into a forest. It creates this high dominance, and sometimes monodominance of longleaf pine, which is the most fire resistant and resilient species in the system. And then also this high density and diversity ground layer, so a savanna sort of architecture to the system with codominance by an understory and overstory layer. The percentage of longleaf pine savanna that remains is one of our typical kind of sad conservation stories. About 3% of this historical range is intact at present. This is a result of conversion of longleaf pine into a variety of other land use types. So they've been converted into agricultural lands, into silvicultural lands. Some cases they've been agriculture and then that has been abandoned and they're now silvicultural lands. Conversion to some human occupation, and then also through fire suppression. And so as I mentioned, fires burn through the system every 1 to 3 years. When fire is suppressed, we get conversion of this open canopy longleaf pine to closed canopy and eventually dominance by hardwood tree species. Oaks, hickories, and other hardwoods will eventually outcompete the pines in the system in the absence of fire disturbance and cause something like this. This is a severely fire suppressed site where we don't think it's been burned for more than a few times in the last 60 to 75 years. And what we can see is that there's virtually no pine left in the overstory. And whereas we have these oaks and hickories that when fire is frequent are shunted into the understory through repeated top killing, and those have been released. They've grown up, the canopy is closed in. This has radically altered the structure of the system, so there's much less light penetration. We can see the difference in the ground layer in this case, where we have a lot more leaf litter showing through. There's a lot more shrubby species. And then in turn, these habitat conditions are wrong for a lot of the native animals. So red-cockaded woodpeckers, gopher tortoises, indigo snakes, and a whole suite of other species would not use this sort of site as habitat. They would abandon a site like this or not colonize it in the first place. There's substantial interest in restoring longleaf pine savannas. Often they start in this closed canopy state. A lot of times from a silvicultural setting, there's widespread planting of longleaf pine trees, loblolly slash pine as well in the southeast, but often managers will focus on longleaf pine plantations, and as I mentioned before, they'll come in with heavy machinery, they'll cut down trees to create the spacing and density that they have set out in terms of their objectives or goals, and they'll start burning the sites. Here you can see some of these longleaf pine trees and see just exactly how fire adapted they are. They're basically the only green left in this site that is recently burned. A lot of this is surrounding management for red-cockaded woodpeckers. They function as a bit of an umbrella species in this system, but there also is a lot of restoration of longleaf pine at a whole system scale for other aspects of the system. So this is maybe a general process. The starting conditions can vary substantially. In particular, some of the sites where restoration takes place may be what we would call remnant longleaf pine habitat. It's never been put to the plow and never converted into agriculture. Whereas in other sites, this might be a post-agricultural longleaf pine stand as a starting point. And we can see just how pervasive this post-agricultural condition is at our research site, the Savannah River site. This is a Department of Energy base. It's about 300 square kilometers, I believe. Sits on the southeastern coastal plain in South Carolina. Historically, we think, based off of some vegetation mapping efforts by Cecil Frost, that this site in the uplands would have been dominated by fire-maintained longleaf. That's the yellow on this map. So this was a fire-frequented landscape. Early settler accounts describe being able to practically see the horizon and only scattered pine trees. They've described how pervasive fire was as well and what they were seeing around. So we think this is what maybe around 1800 or before this landscape looked like. In 1951, the US government took this land. They made it a Department of Energy base. This was part of the Cold War nuclear production efforts. They put a big fence around the outside, and they told everybody, we're happy to pay you for your land, but you're going to have to leave. At that point, this is what this landscape looked like. So the white that you're seeing there are agricultural fields. You can see the sandy soil showing through on this aerial photograph. So what you can see here is that the majority of the uplands on this site had been converted into agriculture at this point. There was a fair bit of forest in the lower lying areas in the floodplains. You can see the floodplain of the Savannah River here, also some major waterways across the site that had substantial hardwood floodplain forests. In the uplands, there were scattered of these remnant longleaf pine patches, some of which are shown in red here. Those are going to be some of the focal areas in the work I'll talk about today. So this is what this site looked like in 1951. At that point, the US Forest Service was brought on board. And so this was Department of Energy land. They brought the US Forest Service in and they said, can you please afforest this landscape? This was owing to concerns about soil conservation. They were watching soils wash away. They were concerned about that. It also had to do with securing this land as a Department of Energy base where they were planning to install nuclear reactors. Individuals might be able to drive across farm fields. They thought it was a more defensible space if there was a dense pine plantation across the landscape, and any vehicle traffic could be confined to the roads or the air. The Forest Service came in, they afforested tens of thousands of acres. At that time, it was the largest reforestation effort in the entire world. And they did a really good job to the point where this is what the landscape looks like, or approximately what it looks like today. So we can see that the agricultural fields have all been reforested. We have a forested landscape that now is comprised of a mixture, a mosaic of remnant longleaf pine and post-agricultural pine plantations, some of which is also longleaf pine. So this presents some interesting restoration opportunities. In a way, we could say the system has been kind of restored. It's been reforested. As I'll describe, though, other aspects of the system differ to this day based off of whether or not a site was in agricultural land use. Now, these sorts of post-agricultural ecosystems, I would argue, represent a substantial restoration opportunity. We, as a human society, have abandoned an incredible amount of agricultural land all over the world. These are some data that were compiled running up through 1990. And at that point, there were 235 million hectares of post-agricultural land in the world. So these are areas that at one point in the past we know were ag fields and then have been abandoned and might be an herbaceous old field like we have plenty of around in Michigan. Maybe these are forested systems now, or something else that was somewhat natural. So these are, again, substantial restoration opportunities. So how much is 235 million hectares? It's kind of hard to grapple with these sorts of numbers. So if we put those in terms of Michigan's, that would be over 9 times the area of our state. If we put all that area together, this would be the 11th biggest country in the world. So a lot of land that we might be able to do restoration on. This also comes with some challenges. These challenges are due to legacies of agricultural land use. So we know that when we do agriculture and then we abandon it, it leaves a signature on that ecosystem. These legacies or these signatures can often be pronounced. Post-agricultural systems very often, if not virtually always, have altered plant community composition. They often have altered forest structure. They have altered soil conditions relative to areas that are, we think, otherwise similar, but just were never in agriculture. These legacies can last a really, really long time. One of my favorite examples comes from southern France, where some individuals were able to find a map that was made by Romans during the period of Roman occupation. So that was over 2,000 years ago. On that map, they found an area that was an agricultural field. Today, it's an ancient forest. If you didn't know better, you would say, primeval forest, looks ancient, the trees are huge. Right next door to that, there actually was an ancient forest, an area that was a forest on the old map. Discussing with people, they figured out that it's always been a forest, at least since then. So they think it was never an agricultural field. So they had two really old forests, one post-ag, one a remnant forest. They measured some things about those. They found that the post-agricultural forest had a different plant community composition and altered soil attributes, even 2,000 plus years after abandonment. And in particular, they found elevated phosphorus in the post-agricultural sites. Now here's kind of the kicker. We don't really understand why these agricultural legacies are often so persistent. Why do they last for decades to centuries to millennia? What are the processes at play? So together what this argues is that we need some more sort of fundamental work to understand the mechanisms that maintain these agricultural legacies if we're going to in turn do restoration on these sites, which seems that in many cases is going to be critical. The authors of this paper from southern France argued that these agricultural legacies were irreversible. And of course, as a restoration ecologist, I say, well, we'll see about that. But we're going to have to figure out our restoration methodologies in order to get restoration right on these sites. So from the perspective of plant communities, we can think about what some of these mechanisms might be. Here is a cartoon of how I think about this. We have our patch of remnant forest. This might be a woodlot, like we see all over our landscape here. There's one on most farm properties. Say it's surrounded by an agricultural field. At some point, we decide to abandon that agriculture, and the ecosystem starts to recover there. Let's say it's a new forest. It may be a relatively old new forest. Maybe it's been around for decades, maybe even a century. The canopy's closed. It's got a forest structure to it, but it's different. We can measure things about that new forest, and it's different, say, in terms of the species of plants that are living in it. So why might that be? Why haven't some of these plants that we see living in this remnant, these relic populations, dispersed and established or reestablished in this post-agricultural forest? So we can break this down and think about some of these mechanisms. So maybe seed production is limiting. Perhaps these relic populations are disturbed in some way. Maybe the system conditions are wrong and those plants just aren't making any seeds. Or maybe they are. Maybe they are making seeds and those seeds are not dispersing for one reason or another. Maybe we've seen local extirpation of a key seed disperser or some other reason that the seeds just simply are not getting out into the new forest. Or maybe they are. And those seeds, when they do arrive, are experiencing incorrect establishment and persistence conditions. I just mentioned that some things about the soils can be really different in post-agricultural lands relative to remnant lands, and maybe that's causing some sort of demographic problem for these species. So maybe the processes of establishment or persistence, forming persistent populations, are just incorrect. So we've been grappling with each of these in turn to try to figure out which, if any, might be at play in our particular system. Okay, so back to the system, longleaf pine. Questions that we're grappling with and that I'll tick through today are, first of all, what are the legacies of agricultural history in the ecosystem? We wanted to describe how remnant sites that we don't think have any history of agriculture compare to post-agricultural sites. All forested at the start of our experiment, but we know they have this difference in history. Secondly, does agricultural history affect restoration? So we can describe all these differences. Does that then in turn cause some of this variation that we know happens when we do restoration? So in other words, do post-agricultural sites turn out differently in important ways relative to remnant sites when we restore them? And then finally, what processes control these agricultural legacies, in particular for understory plants? So in what ways might these various mechanisms that I just talked about be important or not for the reestablishment of relic populations into post-agricultural sites? So again, our system is longleaf pine savannas. These are at the Savannah River site. Just happens to have the same name as the system in the site in South Carolina. What we've done again is we've identified a set of these remnant patches of longleaf pine. We use old aerial photography. We did a bunch of ground surveys. We started with an initial set of 25,000 candidate patches, and through a whole series of screenings, we identified 27 sites that were based off the following criteria. Each one had a patch of what we thought was remnant longleaf pine savanna. So going back to the oldest aerial photography we had, which was taken at the time that the Savannah River site was created, it was a forest. Based off of our ground surveys, the plant composition in particular suggested to us that it had these relic populations and other attributes as well that suggested it was a remnant. Each of these remnant patches then was immediately adjacent to something that we knew was an agricultural field in the past because we could see it on these old aerial photographs. That agricultural field then was abandoned when the Savannah River site was created in 1951, planted with pine trees. We picked sites where they were planted with longleaf pine trees, and those had grown up and closed the canopy by the time we were starting this research project. So these sites had to have all these criteria. We found 27 of them, and we started addressing our research questions within them. So within each of these sites then, we set up a series of plots. These plots were 1 hectare in area, 100 by 100 meters, spanning the 27 replicate experimental sites. There were 126 1 hectare plots that we established for this research study. They were always set up in this way, where they were straddling this land use boundary, where there was one plot within the remnant, and then immediately adjacent to that in the post-agricultural area, there was a second plot. Each of the sites had at least 2 pairs. Some of them had upwards of 6 to 8 pairs, depending on the size of this remnant patch of longleaf pine. Okay, so back to the questions. First of all, we set out to just describe how these areas differ from each other. How do these plots that are in the remnants differ from the post-agricultural plots? So first of all, we can take a look at some pictures. This is a remnant patch of longleaf pine. This is a post-agricultural patch. So we can just see in pictures that the structure of the system is really different. There's different overstory tree composition. There's differences in the understory. We have much more prominence of pine needles, many fewer plants in the post-agricultural sites, broadleaf trees. So this happens because of this particular history of land use where the pine had been selectively harvested, the sites were fire suppressed, this released these suppressed hardwood trees. Those grew up, they closed the canopy, higher light levels then as a result of the light penetrating through the pine needles compared to the broadleaves. So we can see and we described a number of these differences in ecosystem structure based off of whether the site was or was not in historical agriculture. In addition, there were differences in plant community composition. So this will be one first of several ordination plots I'll show you today. Here's the way to interpret these. Each of these dots is representative of one of our plots in the experiment, one of our 1 hectare plots. In this case, we sampled plants along transects in these plots, and this is describing the community composition based off of the identities of those plants. When 2 of these dots are really close to each other, that means that those 2 plots had really similar identities of plants, really similar plant community composition. And when they're really far apart, that meant that the plant communities were really, really different. So here's the takeaway from this. There are really different plant community compositions in the post-agricultural plots compared to the remnants, in spite of the fact that agriculture was abandoned over 60 years ago, and these are immediately adjacent to each other in space. So what this suggests then is that there are a suite of plant species that have failed to recolonize into the post-agricultural sites, and we see that using an indicator species analysis. So this identifies species of plants that are particularly indicative of a particular plot type over the other. In this case, we're comparing the post-ag to the remnant areas. And what we see is that there were about 60 species that were significant indicators of the remnant plots. So they were mostly only found in the remnant areas and not in the post-ag areas. There's also a set of species that are significant indicators of the post-ag areas, and then there's also a set of species that are cosmopolitan. They're found all across the landscape. So we have some good evidence now that there is a lack of reestablishment into the post-ag areas, and this is causing this persistent legacy of former agricultural land use on plant community composition. I described already some of these differences that we see in terms of ecosystem structure. We also see a number of differences in the soils. Some of these result in there being more of something in the soil in the post-ag sites. Just like in that study in France, we saw that there's elevated levels of soil phosphorus in the post-agricultural plots compared to the remnant plots. In other cases, there's less of something in the soil in the post-ag areas. So what we see here is that post-agricultural sites hold water less well compared to remnant sites. We think this is because there's been a loss of organic matter. Well, we know there's less organic matter. We think there's been a loss of organic matter through this history of tillage in the post-agricultural sites. And that organic matter in these highly sandy soils is extremely important for the soil's ability to hold on to water that then might be available to plants. Also very important for the soil's ability to hold on to nitrogen, which is another limiting resource in the system. So we see less nitrogen sitting in the soils in the post-agricultural sites compared to the remnants. So we can describe this whole suite of differences between these sites depending on what their land use history is. We then wanted to know, in what ways does this matter, or does it even matter at all, for the way that restoration plays out in these sites? So to do this, we conducted, in conjunction with the US Forest Service, a thinning experiment, a tree thinning experiment. So remember I mentioned that this is historically a savanna configuration ecosystem, scattered overstory trees, lots of light penetrating down to the understory. Because of this history of selective harvest, tree planting, fire suppression, we started with a closed canopied forest system. So what we wanted to do then was conduct a thinning treatment where some plots were thinned, those are shown as open here, and other plots were left as unthinned controls. We did this in the different combinations so that we had thinned remnants, unthinned remnants, thin post-ag, unthinned post-ag. And these trees were thinned down to very low densities. You can see what this looked like in pictures here. So here's a remnant and a post-ag that's unthinned. I know you already saw some examples of these pictures. And here's what the sites looked like after thinning. So we see that we've kind of reinstated these low tree density conditions. We've at least structurally restored the savanna to these sites. I should mention also in terms of color coding, I'm going to show a bunch of graphs now. The blue is always going to be remnant. The red is always going to be the post-ag. And open symbols are going to be the thinned plots. Closed symbols are going to be the unthinned control plots. So the question then is, what are the effects of restoration thinning in this system? And does it matter if we do this thinning on a post-ag area relative to in a remnant stand? So looking first at species richness, so this is the richness, the total number of understory plant species that we observed along transects. These transects were a 1/10 hectare in area, 10 by 100 meters. What we can see is that in unthinned plots, statistically there's no difference. So remnant areas, though qualitatively slightly higher, statistically are supporting comparable levels of species richness to the unthinned post-ag areas. This is at the 1/10 hectare scale. We see very similar results looking at plot sizes at the 100 square meter and 1 square meter scale. So the question is, what happens now when we do the thinning treatment? We know in this system that there's strong asymmetric competition between trees and understory plants. When we have increased numbers of overstory trees, we see reductions in understory plant species diversity. And that's exactly what we see here. We see a 27% increase in understory plant species richness. So an increase of about 80 to about 100 species of plants per 1/10 hectare. To brag a little bit about my ecosystem, not that I really have any right to brag about an ecosystem, but these levels of diversity are fantastic. 100 species of plants. And we've recorded, I think, over 400 different species of plants across this system. So really, really biodiverse, fun system to work in. Key points here are that when we do the restoration thinning, species richness increases, but it does so in the same sort of way in remnant and post-agricultural sites. So again, statistically, these differences are not different from one another. So we see the same sort of effects of restoration in remnants and post-ag areas for species richness. The same is not true for community composition. So remember, I showed you the ordination before, illustrating that community composition of understory plants was really different before we started the restoration process in remnant and post-ag areas. So what we're seeing then is that following thinning, we still see a difference between remnant and post-ag, but those communities in turn are different from the unthinned sites. So as a consequence, we essentially have 4 different sorts of plant communities that have resulted from our experiment based off of the combinations of remnant and post-agricultural land use and thinning or unthinned conditions. So what this suggests is that thinning has caused the colonization of a whole suite of new plant species into the system, many of which may be more ruderal in nature, but this is at least as suggestive of the fact that this hasn't caused these relic populations to reestablish into the post-agricultural areas, because if that happened, we would have seen these plots coming together after we did thinning, and we don't see any evidence of that statistically. Okay, so that brings us to this third question then. What processes control agricultural legacies for understory plants? So we see this persistent legacy of agricultural land use for understory plant communities. We've done restoration and it doesn't seem to get rid of that. We think it's because there are a whole set of plant species that have not spread into the post-ag areas, and when we do restoration, they still haven't spread. So we focused on a set of these species now. We focused particularly on a dozen of these remnant indicator species that are mostly only found in the remnant plots, and then we ticked through these hypotheses. Remember what they are. Maybe they're not producing seeds. Maybe what seeds they produce are not dispersing into the post-ag areas, and maybe any seeds that do disperse are not establishing for one reason or another. Perhaps the environmental conditions are wrong in the post-ag areas. So to show you some data, this is, I think, was the first graph we ever created, at least I've ever created in my life, that I said, I don't need to run stats on this. What we found was that when we did the thinning treatment, plant reproduction increased by an average 6,500%. So what this shows is that those dense overstory conditions were causing very low levels of reproduction. This is in terms of flower production. For a subset of these species, each of these is a different species that we followed. For a subset of these species, we also tracked seed production. We saw very similar patterns for those species. So these dense overstory conditions make sense, are not right for reproduction of these understory plants for whom savanna conditions are correct habitat. Okay, so that seems to support this first hypothesis. Maybe there's limited seed production in these remnant areas. How about seed dispersal? So seed dispersal is a notoriously difficult thing to study. Seeds are usually little. The ones that go a long ways are often disproportionately important for ecological and evolutionary processes, and we run into a needle in the haystack problem. It's very hard to relocate seeds that have gone long distance. We've, by some methods, including stable isotope marking, have tried to tackle that problem in my lab. For the purposes of this study, we took a different approach. We said, we're going to become the seed dispersers. So we collected seeds of a whole bunch of these species, and then we sowed them into the post-agricultural areas. Our logic was then that if they established, that would suggest that dispersal is limiting. If we sowed them and they were unable to establish, that would be suggestive of some inappropriate environmental conditions. So this is what this looked like. Again, we had this experimental setup. All of this is replicated 27 times. We had plots where we added seeds of 12 of these remnant indicator species. We did this in remnants. We did this in post-ag. We did this in areas that were thinned or unthinned, and we compared those to nearby areas that were unseeded controls. So again, if we see, for example, that these species are establishing here, but not here, that would be evidence of limited seed arrival, lack of seed dispersal. If they're establishing more here than here, that would be suggestive that the environmental conditions, because again of these dense overstory conditions, are inappropriate for the establishment of these species. Now, finally, we logicked that if establishment is high here in the remnants and low in the post-agricultural areas, it was something else, the soils, or the list could go on, that whereby the environmental conditions were wrong in the post-ag areas. So here's what we found. First of all, over 99% of the occurrences of these focal 12 species were in seed addition plots. These are just in the post-ag areas. So this is really good evidence that seed arrival, that process of seed dispersal from the remnants to the post-ag areas, is extremely limiting for the reestablishment of those populations. Moreover, the rates of establishment, the species richness, or very similar results if we look at the counts of individuals across these sown species, was much higher, in this case 125 or so % higher, when we added the seeds in thinned plots compared to when we added the seeds in unthinned plots. So together what this suggests is that seed dispersal is limiting, and environmental conditions, again because of these dense overstory conditions in the post-ag areas, is limiting the establishment, and at least so far, 3 years into this experiment, persistence, as these populations have been persistent over time. So this is, again, just looking at the post-agricultural areas. What about that comparison between the post-ag plots where we added seeds and the remnant plots? So maybe we're seeing higher rates of establishment here, but these are still minuscule compared to those rates of establishment in the remnant plots. So to do that, we've looked at then the focal species richness and again the abundance in these plots where we sowed seeds in the remnants and in the post-ag areas, and what we see is no evidence for that idea. So the rates of establishment are actually about 50 to 60% higher in the post-ag areas compared to in the remnants. So what this suggests is that in spite of all these differences that we're able to measure between the post-ag and the remnant areas, this is not suppressing reestablishment of these populations. And if anything, these post-agricultural areas are slightly better than the remnant areas for the reestablishment of these populations. So putting this together then, what we see is that we see support for the hypothesis of limited seed production, reproduction, that can then cause any export in the first place from remnants. We see very limited dispersal in this system. We think that that's a very major cause of limited recovery. Excuse me. But we don't see any evidence that the environmental conditions are just wrong. It's not wrong habitat for these species in the post-ag areas. So we've been doing restoration. We've thinned the canopies. We've started experimentally burning these sites. We've waited about 4 or 5 years. So what do we expect? We expect then for some of these relic populations to start spreading into the post-agricultural areas, and particularly when we did the thinning treatment. So the question is, has restoration caused spread? And the answer is, to date, no. So what this is showing is the richness of remnant indicator species, the number of these indicator species per 10 by 10 meter plot, 100 square meter plot, at distance from that boundary between the remnant and the post-agricultural plot. So what do we see here? Well, first of all, there are many more of these indicators in the remnants. That is not too surprising, and actually based off of the way we defined indicators, bound to be the case compared to in the post-ag areas. We see that there has been some spread of these species into the post-ag areas. So we see elevated richness of those indicators for maybe about 30 meters into the post-ag areas. But that amount of spread is virtually identical, certainly not statistically different for areas where we did the thinning or not. So what this has shown is that several years into this experiment, our restoration thinning has not caused elevated rates of spread. If that was the case, we would have seen that bar up higher here. Okay, so I want to tell you a little bit about some of the ongoing work with restoration for pollinators now before I wrap up. So with this, I'll return back to the second question. Does agricultural history affect restoration? Talked already about how that seems to be the case for plants. How about for pollinating insects and for the process of pollination? So to study this, we've sampled pollinators. So far, we've specifically focused on bees as important pollinators. We're also focusing on other pollinator groups. within these different sorts of plot types. Again, the remnants, the post-ag, thinned and unthinned. For bees, we've used a combination of passive sampling approaches. We use these things called bee bowls that are painted different colors. The bees think they look like a flower. They come and land in them to check out. There's soap in the water. When the bee lands, it breaks the surface tension and it drowns, and we just sort of passively sample bees this way. We've coupled this with active netting of bees along transects in the site. So we can compare then the bee community in these different plot types. We've also done a sentinel plant pollination experiment. This was our first foray into this. We used a model species. It's a mustard species, not a native member of this ecosystem. It is, however, an outcrosser. And so our logic was we could stick these out into the different plot types. Some are bagged, some are unbagged. And if the different plot types are affecting pollination differently, we would see higher rates of fruit set in those plants that are sort of sampling the pollinator community in one plot type over the other. They were deployed for just a few days, then they were retrieved. They sat in greenhouses, we let them develop fruit, and then we counted the fruits on these plants. So here's what we found in a nutshell so far. This is ongoing work. This is being led up by Sabri Breland, who's our lead technician on the project. What we found is that restoration thinning increases I'll show you first the abundance of bees in these plots. So we see a pretty substantial increase in abundance, about 170% more bees in plots that receive the thinning treatment compared to the unthinned controls. The land use history, agricultural history does not matter here, and there's no interaction between agricultural history and the thinning treatment. Very similar patterns when we look at species richness and diversity. Okay, so thinning matters. Agricultural history does not matter for diversity of bees. Actually fairly similar to what we've seen in terms of plants. How about composition? Composition is a little bit of a different story. So you can maybe immediately recognize, well, this is another of these ordination plots. This one looks like a mess compared to the other ordination plots for plants. And the reason is that there's much less difference in pollinator and bee community composition across the plot types compared to for plants. And in fact, there's no effect of agricultural history on bee community composition. There is a significant effect of overstory thinning, but it only explains 5% of the variation in bee community composition. So there seems to be a pretty big difference in the ways that bees are responding to agricultural history or to restoration compared to plants. How about pollination? So what we found was, first of all, our experimental system we think was a good one in that bagged plants had about 33% less reproductive output compared to the unbagged plants. So access to pollinators mattered, but it worked the same way across the remnant and the post-ag, unthinned and thinned. So we can see there's some qualitative wiggling there, but statistically, none of these are different from each other. So what we see is that, at least based off of this first hit on this question of how restoration and agricultural history affect pollination, the answer seems to be that pollination rates are comparable across these plot types, which was actually a big surprise given that we saw that huge increase in bee abundance and bee diversity. We're following up on all this work this season. We're working to work more with the native plants in the system. Our approach is to construct interaction networks where we are visiting plants and we're recording the species of all the pollinators, bees and others, that are visiting individual plant species and doing it across all the different plot types. So we can ask about who the visitors are and what the rates of visitation are on the native plant species in this system. Okay, so let's wrap things up. What have we learned here? Revisiting the questions. What are the legacies of agricultural history on this ecosystem? We see a whole suite of them spanning soils, aspects of ecosystem structure, plant composition, to name a few. So the question then was, does this matter for the way that restoration plays out? When we do restoration on the post-ag and the remnant areas, do they differ? For plants, the answer is yes. And what we're seeing is that these legacies of agricultural land use essentially set the template for how restoration plays out. Restoration works differently for plants on remnants versus post-agricultural sites, and this doesn't seem to be the case for bees and for pollination. Our hypothesis is that organism mobility may be important here, where the plants we see good evidence for dispersal limitation, whereas bees are well known to be very, very strong movers across landscapes. They may also perhaps be more generalist in who they're visiting in terms of plants, so maybe some of those plant community composition differences don't matter so much for them, something we're again following up on this year. And then we wanted to know what processes control agricultural legacies for understory plants. We see that seed production, dispersal from remnants, and then also these dense overstory conditions are important for this lack of recovery. In turn, through restoration, because we've affected a number of these, we expect to be able to cause spread of these relic populations. We just haven't seen that yet, and we're continuing to monitor these plots. So finally, what's this mean for restoration? So we're working with the US Forest Service on this project. They, of course, are very interested in understanding how they can restore this land that they have, recognizing that it's been impacted by a variety of land uses over the years. So first of all, we see that tree thinning is extremely important for the way that restoration works. All the land managers in the whole region know this. We're not going to teach them anything new. We can provide some data that can help back up what they already know. What we're showing is that site selection, however, is extremely important. The general paradigm is that land managers do not typically consider the agricultural history on sites when they pursue restoration. In part, this is because they're often restoring around this woodpecker species. What we're showing is that those decisions do matter for plant aspects of the system. We're showing that seeding within the system may be important. Compared to in the Midwest, where, for example, in prairie ecosystems, seeding is an integral part of the restoration process, it's not widely employed in the Southeast, and our results show that seeding may be a beneficial restoration practice, particularly when it can get coupled with overstory tree thinning and presumably fire as well. And finally, there may be an important role of passive restoration. Targeted thinning, particularly of remnants, but also perhaps of post-agricultural areas, may cause passive spread of these relic populations that may be conservation targets. And the big question now for us is, over what sort of timescales might this take place? Are we going to see this over the life of this experiment, which was slated to run for about a decade, or is this well beyond our lifetime? We'll see. We'll see what we can learn by the end of the experiment. With that, I'll say thanks, answer any questions that we might have time for.